the fossa, the spoiler is the pterygoid venous plexus. Remember, the spoiler during the process of dissection is pterygoid venous plexus. That pterygoid venous plexus is also spoiler from the point of view of clinical anatomy. ঠিক আছে? Pterygoid venous plexus, about the pterygoid venous plexus, I do not know how much you will get in your Indian textbooks. But remember, pterygoid venous plexus, what I have said
कनेक्शन अभी आपको याद नहीं है.
Please be honest to raise your hands whether biochemistry is an interesting subject at all or not. Please be honest, because I am not going to count. I will see your face. Only I want to know the hands up. How many of you are really find interest in reading biochemistry? So no. So that's a handicap. So what I would like to say, that there are many handicaps. It's a mental block. It's a mental handicap, not a physical one. You have to cultivate the land of medical science. You have to find your own technique, how to cultivate. Nobody else will teach you. Just a video. Please watch this video. See how one can cultivate. इसको कैंसिल करो क्लोज करो खत्म कर देना. Wait. See, everybody is a teacher, everything is a lesson, and it's a good lesson for me. I mean, when I saw the video, it drew my attention like anything. You see, this is an old tribal farmer, no way to earn livelihood, neither to purchase. Does it stimulate you in any way to read now biochemistry? Can you imagine? I think reading biochemistry is not that difficult, like the farmer. Video the bond. Close go. 16. Open. Hello, what is this structure? What is this structure? Please interact. What is this structure? Am I going to teach you histology? Histopathology? What is the structure? Can anybody tell what is the structure? You have not seen histology? You have not seen histology? None of you have seen any slide. Strange. See, the left hand side picture is histology of the skin. You can see the stratum corneum on the top. Okay, you are the one that. See, this is the stratum corneum and this is the squamous cell layer and this is the structure of intestinal villi. So what all you have read so far is the metabolism or anything going within the cell. The cells are like the bricks of a multi-storied building or even any building or any house. Only brick will not constitute any house and make it strong and function it properly. So apart from these cells, there are many things around us, around that cells. What are these? What are these? These are not cellular elements. These are not cellular elements at all. And the structure is wildly different. See, the multi-storied building has many rooms and each room has a different function for which the interior, decoration, everything is different. Same is true for and much more complex in our body. All our tissues are not similar, so therefore you will not find the same structure everywhere. If I go a little bit deeper, you see this is the epithelial cell and there is a basement membrane of the epithelial cell and the capillary also, and this is the space, and this is the cellular membrane which you have seen so far. This is the transmembrane protein, that is integrin, and this is microfilaments within the cytoskeleton. But outside this cell, there are many more biomolecules like proteoglycans, collagens. Can you next? এটা কাজ করছে না, এটা কাজ করছে না. If you go further down, see this is the plasma membrane, and these are the microfilaments which I have shown, and again the collagen fibers, bigger, still bigger, and this is the proteoglycan complex, which is having a polysaccharide and to which proteins and carbohydrates are attached. And further, if we enlarge this picture, you will see the extracellular membrane, matrix, the basement membrane, and the cytoskeleton. So in this structure, extracellular one, you can see the collagen, elastin, fibronectin, laminin, integrin, it is a transmembrane protein, microfilaments, proteoglycans, and hyaluronic acid. Next. If you look further deeper and enlarge the picture, you will get an interstitial matrix and the basement membrane. Both constitutes extracellular matrix, and that is a cellular membrane, and this is a cytoskeleton. And again there is elastic fiber, proteoglycan, and the fibrillar collagen and non-fibrillar collagen, nitrogen and laminin. Next. So today we are going to discuss basically our extracellular matrix, which is there in your second paper. So it is very difficult to cover extracellular matrix in one class. So I have decided that I will cover the main part first, and tomorrow in the morning class I will cover the rest of the things. Otherwise it will not be possible for you to digest. What I would suggest that today, after going from the class, you go through the chapter, otherwise you will not be able to read, and your time is very less because exams are ahead. So today we are going to discuss extracellular matrix. Please try to remember many things. I will repeat several times, so that instead of gossiping inside amongst yourself, please concentrate here. Otherwise you will neither be able to write anything nor will be able to say anything before the examiner. I am sorry that you still are gossiping. It's very bad. So this is the most important slide. You must remember this thing oft repeatedly in the entire class. I will repeat the same thing. So this is the extracellular matrix, that is the non-cellular component of all tissues and organs. And it is having two roles. One is structural and another is functional. Its structural role is the supporting structure and forming a network, just like your bricks. Its functional part, that is the most important part. Each and every component which I have said so far, fibronectin, laminin, collagen, they do, they are really involved in the development and differentiation and maintenance of tissue and organs, and they communicate between the interior of the cell and outside of the cell, so that the cell health is maintained, and they are responsible for biochemical and functional properties of each organ. This part you must be able to remember, and you must be able to write what are the functions of extracellular proteins. That is the most important part. They also play a role in inflammatory states which I will discuss, spread of cancer and disease development. Now, as I told you, in a multi-story building, all the rooms are not same. In our body, all the organs are not same. So the extracellular matrix of each organ will also not be same, because their functions are different. So according to that extracellular matrix is formed and they perform different functions. So its major components are three basically. One is structural protein that forms the basis, structure of that organ, and then specialized proteins which functions, special functions, these functions, and the proteoglycans. And of course water must be there, because that maintains hydration of the entire extracellular matrix. So the major components you must remember at least these 6 points. You may not be able to tell other points which I will discuss. One is a structural protein, that is collagen. That is the most abundant protein, about which I will discuss at the end. Elastin and fibrillin. A specialized protein is the fibronectin and laminin. They form a mesh of fibers. And proteoglycans I will cover tomorrow. Next. So elastin, as the name suggests, this is elastic in nature. So it's a stretchy protein like a rubber band, and it can stretch, it can contract, it can relax. And there are organs in our body like lungs. During inspiration and expiration, there is a movement, blood vessels also expand. So these elastin tissues are present in the lung. Urinary bladder, you know that when the bladder is full, you feel the urge of urination and then bladder contracts. So that is essentially elastic tissue. So some ligaments are there and the blood vessels, particularly the aorta, because that expands and relaxes during systole and diastole. So they are formed of elastin tissue. If we look a little detail that in the histologically, that collagen, elastin, they interact, and if we look a little deeper, you can see that tropoelastin is formed, from which elastin fragments are formed, that they are getting cross-linked, and then elastase enzyme basically breaks down the cross-linking, and finally they are degraded. So they are major components of tissues requiring stretchiness, as I told you, like your lungs, bladder, blood vessels. They are tightly associated with collagen fibers that I have shown in the histological structure. And 1/3 of the residue is glycine, and a massive amount of proline and alanine is present. One must remember that this maturing elastin has a desmosine cross-link. This desmosine cross-link is having 4 lysine residues, and this is copper is essential for cross-linking. Otherwise, if there is copper deficiency, there will not be proper cross-linking, and this will not function properly. And one thing you must remember, at the end in one slide, I will show you the difference between collagen and elastin. That these four components which are typically present in collagen are absent here in elastin. That is glycine XY sequence that is unique for collagen, hydroxylysine, a glycosylated hydroxylysine, and triple helical structure is unique for collagen, but all these are absent in elastin. Next. So fibrillin, you can see both the structures. I have shown it in detail. So there is a fibrin microfibril, and it is formed in both elastic tissue and non-elastic tissue also. It's a major structural component of microfibril, and they form basically a scaffold. Scaffold means it's a structure for deposition of elastin secreted from fibroblast. Actually, fibroblast secretes the majority of the extracellular proteins, and found in elastic fibers, also in elastic free bundles, like here I have shown you that non-elastic tissues it is also found. And fibrillin 1 is the major component. You must remember, among the three, fibrillin 1 is the major component, and if there is a defect in the fibrillin 1, it causes Marfan syndrome, and I will show you the image and features of Marfan syndrome at the end. Next, এটা কাজ করছে না, এটা ব্যাটারি বোধহয় খারাপ আছে. Another is fibronectin. You see, there are four domains. If you remain there, I am unable to see from here. So what you will see there? There are four domains which can bind fibrin, collagen, and cells and heparin. So if you look here, that extracellular matrix is composed of basement membrane and intracellular matrix, interstitial matrix, and a number of proteins are present, one of which is fibronectin. The structure is here. So if you look at the confocal microscope, if you look at, stain it, it will stain like this, and it is basically composed of a dimeric structure where two disulfide bonds are connected at the carboxyl terminal. So they bind to the membrane sparing receptor called integrin, which I will show in the next slide in a better way. And ECM proteins like collagen, fibrin, and heparin sulfate. Again, what I told in the function, that's a key player in the communication between intracellular and extracellular matrix. The major function of extracellular matrix is to communicate between the cell, inside the cell and outside the cell, so the health of the cell is maintained and it can function optimally. So it is basically forming an insoluble network separating and supporting the structural tissue, and it plays an important role in wound healing. Next slide. Next. Interaction of fibronectin. Now see, as I told you, that it interacts with collagen, it interacts with laminin. These are the major proteins of extracellular matrix. This I repeatedly I told you the second slide. I am telling you again, at least these three names you must be able to tell. And this is attached, fibronectin is attached to integrin, receptor, that is a transmembrane receptor, and with actin it interacts through four attachment proteins, and it is anchoring the cells to the ECM and intracellular and extracellular communications. So laminin is another one. It's a triple helical structure. It's a network-like structure composed of alpha, beta, and gamma chain. And if you look at it, that laminin is attached to the epithelial cell by integrin and the destroglycan, type 4 glycogen. It's a major component of basal lamina. Basal lamina means basal membrane, one of the layers of the basement membrane. It's a protein network found in the most cells and organs, and it interacts with the plasma receptor adjacent to the basement membrane. And again, the same function which I told you earlier, cell differentiation, migration, and adhesion. If you look at the confocal microscope, a corneal cell, there are 5 corneal cells which are being shown in magenta color, and the green color is the structure of the laminin. Next. Now, one important thing is that glomerular filtration. You have seen that the urine is formed by the glomerulus. I mean, plasma is filtered, but it doesn't allow larger molecules to pass through, only smaller molecules and water, because it is having a pore size. And if you look closely, that there is a capillary side which forms an endothelium, and the Bowman's capsule side, that is an epithelial layer, and there is a slit pore, filtration slit. But glomerular basement membrane is having also a pore size of only 8 nanometer across. So it can allow only small molecular weight like inulin and water, not larger molecules. But albumin size is 3.8 nanometer, it is still less, but still it cannot pass through. The major reason is that it is prevented to pass through because negative charge of the albumin ion. Why albumin is negatively charged in the blood? What is the isoelectric pH of albumin? Can anybody tell what is the isoelectric pH of albumin? Nobody is bothered hearing, nobody is listening. What I am talking, I don't know. I mean, everywhere, everybody is pursuing with each other. It's a sorry state of affairs. When you don't know what is the isoelectric point and what I am talking, you are not understanding at all. See, the isoelectric point of albumin is 4.7 and the blood pH is 7.4. So at alkaline pH, the albumin will be negatively charged. And because of the negatively charged, this heparan sulfate and sialic acid containing negative charge, they don't allow, they repel basically. So therefore, the albumin cannot pass through. That is one of the major reasons that most of the persons do not know this aspect. So despite being a smaller diameter size, it is not allowed to pass through, but definitely a small amount pass through, because there is continuous wear and tear going on everywhere in the tissue, and through that thing it passes through. Okay? Next. So contractile protein, muscles you have seen, actin and myosin you have seen, running, walking, jumping, even gymnastics. Movement of cell, one is diapedesis of the WBC. When there is any inflammation at any site, usually there is cellular inflammation, and the space endothelial, the space between endothelial cells of the capillary that gets widened, through which the WBC squeezes out. And the WBC also contains some contractile protein that is called actin and myosin, and they passes through these pores into the extracellular space to kill the bacteria or virus or pathogenic organisms. So they are also having contractile protein. Movement of sperm. One of the most important things, see, size of the sperm is only 55 to 60 micrometer, but the speed is 5 millimeter per minute in the uterine cavity, and in a matter of several minutes, it reaches from the posterior vaginal, fornix to the ovary through the protein called tubulin and dynein. Now, can I have the video? Now we see how the sperm moves. That tubulin and dynein is such a small structure, such a small protein, contractile protein. ব্রাউজারে পেস্ট করুন, পেস্ট, হ্যাঁ সার্চ দিন, এন্টার দিন, এন্টার টিপুন, হ্যাঁ জাস্ট সি, এনলার্জ করুন, এনলার্জ করুন। See, this is a small protein. See, you don't, if you want to find interest in biochemistry, you have to go through these slides. These videos, these were not available during our time. So a small protein, contractile protein, can you see, imagine the degree of activity of a small contractile protein. Close to main slide, main slide back. It is close. Ever slide it চলে আসুন, slide আসুন, slide there. Close করুন। এখানে করুন, এখানে শর্টার দিন, শর্টার দিন, শর্টার দিন, শর্টার শর্টার দিন, শর্টার দিন, শর্টার দিন, আমি দেখতে পাবো, শর্টার দিন, আরো আগে, আরো আগে, শর্টার দিন, শর্টার দিন, এইখানে, এখানে, এটা এটা এরপর না, হ্যাঁ এরপর, হ্যাঁ তারপরটা, তারপরটা যান, তারপরটা যান, যান, পরটা যান, পরটা, পরটা, পরটা। So now you see, if this dynein and tubulin they are absent, that will not cause the sperm to move properly, and that is the reason why there will be immotile sperm, or even the cilia will not function properly, because cilia also requires movement. Whenever some foreign body enters into the nose or into the throat, there is a ciliary movement. Because of the ciliary movement, these foreign bodies are thrown out. So if you do not have the cilia properly, so therefore you will be having a chronic sinusitis, and also lung infection, that is bronchiectasis. And this you will read in the later class, that is called Cartagener's syndrome. These three constitutes situs inversus, means the opposite side of the structure should be present in the opposite side of the body. And there are secretory proteins which are expelled through exocytosis or entered into the cell, that is phagocytosis. That requires also contractile protein, otherwise you cannot expel it. So these tubulin, actin, microfilament, kinesin and intermediate filaments, they are all involved in this exocytosis or phagocytosis process. Next. So keratin is present in the hair, skin, nail, horn and hoof. So the main structure of fibers is alpha keratin, and the matrix is produced by the keratohyalin. There is a disulfide bond, it is present in the alpha helical structure. The keratohyalin is forming the matrix. It's a cysteine-rich polypeptide chain held together by disulfide bond, as I showed you. And there is a more bond, means more harder. You know that the hair, hoof or nail of every person is not same. I mean, the characteristic quality is not the same. It is because these keratins are not, this keratohyalin is not that much, is not uniform in all the individuals. There is a variation, and because 8 types, as I mentioned in the list, of collagens are made up of 30 different polypeptide chains. So there are 30 different genes. Can you can you make less noise in the class? Otherwise, you please go out from the class. Do not destroy this class. If you don't like, please go out from the class. Don't make noise. It irritates others who are listening. And I am repeatedly telling that collagen is the most important thing, and you may be asked, you will be definitely asked about extracellular matrix on collagen. You are not hearing, you will not read also after going out from the class. So there are 28 different types of collagen, and I have given the list. And there are non-collagens also, because they are having a collagen-like domain. And it may be homotrimeric or heterotrimeric. Homotrimeric means it is having 3 identical chains. And heterotrimeric, if there is a chains are different. Like type 1 collagen is having 2 pro-alpha chain and 1 pro-2 chain. That means it is not homotrimeric. It is heterotrimeric, because the chains are different. If it would have been 3 structures, same thing, then it would have been homotrimeric structure. So the most of the collagens and other extracellular proteins are secreted by the fibroblast, and this function of the collagens are very important. They support to organs and providing tensile strength. They regulate cell adhesion, alignment of cells for anchoring, support chemotaxis and migration, and direct tissue development, helps with differentiation and proliferation. So when there is an injury in the blood vessels, actually the collagen is exposed, to which the platelets adhere, and it causes plaque formation, and then the bleeding stops. Next. So the structure of the collagen is most important. Again, it's a triple helical structure, 3 polypeptide chains, usually alpha chains. See, this is the glycine at every 3rd position. At every 3rd position, glycine XY, glycine XY. And if you look at this alpha chain, glycine is on the top. Every 3rd amino acid is a glycine structure. The entire collagen type 1 is triple helix, others only in a fraction. And if you look at the thing, it is a triple helical structure. And you must remember the quarter staggered alignment. That is, this neighboring collagen is slightly less than 1/4 of the other collagen. See, this quarter staggered alignment is the most important thing. And is displaced longitudinally from its neighbor by slightly less than 1/4 of its length. You can see there is a gap and there is a less than 1/4. It's a rod-like structure, and you can see this is the structure. I will go to the next slide, it will be clear. So, proline and hydroxyproline, they confer rigidity, and there is an extensive post-translational hydroxylation. Proline is hydroxylated by prolyl hydroxylase, and lysine at one position by lysyl hydroxylase. So, cofactors vitamin C and alpha ketoglutarate is required. Collagen fibers are stabilized by cross-linking by lysyl oxidase. So that confers tensile strength. It's a rod-like structure. Fibrils associate to form fibers, and tendons, fiber associate to form bundles, and these 3 types of collagen are maximally present. So if there is a wound anywhere during treatment purpose, you must remember that collagen formation is essential, and epithelialization of the cell. So since this I read in biochemistry as a routine, although it was not told in any of our clinical classes, but I give vitamin A and vitamin C as a routine, those who suffer from injury and those who undergo operation, because that requires healing. The healing is at a faster rate than normal if you give adequate amount of vitamin C, because it will help in the synthesis of collagen, and also vitamin A because it causes epithelialization. And there are a number of micro minerals which are required. So in fact, there is one nurse who was having diabetic patient who was having extensive burn on the both hands, and it required skin grafting. It was a difficult task. She came to me. I gave her advice only vitamin A, vitamin C, and micro minerals. Minerals only, the tablet. These 3 tablets within 15 days time, both the hands become normal, and you cannot differentiate now that whether it is a burned hand or it is a normal hand. So that is the importance of knowing this thing, that basis of biochemistry is that treatment. You can design many more treatments if you know the basic biochemistry of each and everything. Next. So collagen undergoes extensive post-translational modification before becoming a mature collagen. Pre-pro-collagen is formed, and then pro-collagen from hydroxylation of lysine and glycosylation of hydroxylase. And then basically after entering into the endoplasmic reticulum, it gets released from the Golgi apparatus, and extracellularly tropocollagen triple helix structure is formed. This is formed within the crypts basically, not exactly extracellularly, and then it is clipped by peptide bond because the terminal peptide. Can I now request you finally to leave this hall? Please do not make noise. Please do not make noise. If you want to make noise, please go out of the class. At least there are 5 students who are listening. Let them listen. You can go out, I don't mind for you. If a single student remains present, I'll teach him or her. I mean, he will neither hear nor he will allow the others to hear. What is this thing going on? I don't know. HOD rebuked you like anything in the class. And again, this will be recorded and everything will be heard by him. And he will face the consequences during next classes. And can't you learn the discipline also? If you don't like the class, please don't come. Don't waste my time also. Don't waste others. It irritates me. I mean, there are many important things which you have to learn. You cannot leave. You have entered the medical discipline. You have to be a doctor one day. You have to behave properly. How many times I will tell you? I didn't tell you in the class that you will be a doctor in future. Your behavior must be proper. In next slides, I will be showing you the clinical pictures which you will see in the OPDs. But you will not be able to diagnose it properly if you don't hear this thing. And at that time, you will not be able to find out a clue. What is the cause? You will not be able to have a suspicion also. What is this disease? Destroy the mode. Simply have destroyed, flow of the class also. Next. So the collagen undergoes extensive post-translational modification. There are some intracellular and some extracellular. There is a chart in the book. From the book you can read. It is just like insulin, that pre-pro collagen, that cleavage of the signal peptides, and then hydroxylation and disulfide bonds. And the extracellularly, there are several things which you will read it. Next. Some collagens, remember, do not form fibrils. See, there are a group of proteins which do not, the collagens which do not form fibrils. As I told you earlier, from collagen you will get the microfibrils, but some collagens do not form fibrils because they are characterized by interaction of the triple helix. That glycine XY sequence will be absent there, and that globular structure will be interspersed. Network like collagen form the network of basement membrane, And there is one thing you must remember also, that fibril associated collagen with interrupted triple helix. You just remember this thing. You may not be required to remember all these things, but at least laminin, fibronectin, you must be able to tell. Next slide. Next. So difference between collagen and elastin, this is another important slide you must remember. So collagen, there are many different genes. I have told you there are 30 genes, and there are 28 types. But for elastin, there is one genetic type. Triple helix is present here. There is no triple helix. Glycine XY sequence is very unique for collagen, which is not present. Presence of hydroxylation is there. There is no hydroxylation. Carbohydrate containing, there is no carbohydrate. These are the essential differences between the collagen and elastin. And intramolecular aldol cross reaction, intramolecular desmosine. I have shown you there are 4 lysine residues which form desmosine cross linking, and presence of extension peptides during biosynthesis which are cleaved. This is not, there is no extension peptide. Next. So collagen biosynthesis is basically a very complex procedure, and more than 30 genes are involved, and at least 8 post-translational enzyme catalyzed steps are there. So since 30 genes are involved, it is quite possible that there will be mutation and that will cause disease. Okay, next. So if you look at the collagen genes are actually designated. There is a, international almost, way how to express it. That collagen means this, you write collagen in within italics in capital letter, then type in Arabic numerals. Here you can see collagen there, and then A, and then number of encoding through alpha chain. So type 1 collagen, as I told you in the earlier, that it is having COL1A1 and COLA1A2. That is the gene for pro alpha 1 and alpha 2 chain. As I told you earlier, there are 2 pro alpha chain and 1 pro alpha 2 chain. That's why it was a heterotrimeric structure. And type 2 collagen, that is the gene for this is a homotrimeric collagen. And see one disease which the picture which I will show it to you, that is Ehlers-Danlos syndrome. You can see there are several genes, but Ehlers-Danlos syndrome is found in defects with all genes. You will find many genetic diseases in which the defect will be there. There will be genotypic defect, but phenotype will not be the same. But here there are different genes which are involved in different types, and this is because of gene-gene interaction, genetic heterogeneity, and many other things. Like thalassemia, you know that it is a defective gene, but just like thyroid, which I showed you, the thalassemia syndrome book will weigh nearly 4 to 5 kg. So therefore, that's a big book. So genetic heterogeneity, gene-gene interaction are most important things. Genotype and phenotype will also not match. Neither, and the reverse is also true. Next. So these are the 6 different types, but out of which class type 1, type 2 and type 3 are most common. Now diseases due to defects in collagen, which you will see, and you have to remember at least the biochemical part, not the clinical features which I will show you. Next. So you just remember the upper part. Osteogenesis imperfecta. This is due to type 1 collagen, and there is a single defect, that is replacement of single glycine residue by cysteine, and you will get a picture like this. A baby who is born with a defective spine, that is scoliosis. Can you see the spine is not proper? So disruption, which is because of the disruption of the triple helix near the carboxy terminal. and there is no fibrillar array, and there is brittle bone. So therefore, there will be multiple fractures and skeletal deformities. Can you see? If you see this case in the OPD, you will not have a little bit of doubt that it can be due to a disease of the ECM until and unless you know this thing. And once you find this thing, you will try to find out the cause, and you will not be able to really identify. And that is the importance. At this stage, you must remember the disease and the defect, not the other part. I have showed it to you only to make the subject interesting, nothing more than that. Next. So this is Ehlers-Danlos syndrome. A defective type 3 collagen due to defective lysyl oxidase, because that stabilizes the proline and the hydrolysis. There is weakening of the collagen, and there is hyperextensibility of the skin. If you drag your skin like this, it will not be dragged. You cannot stretch the skin like this. If you want to try to touch your tongue with on the tip of the neck, you cannot. You cannot make your fingers like this. There is hyperextensibility. Can you see the hyperextensibility? So this is a unique feature. Once you see this thing, you have a little bit of doubt that this might be a case of Ehlers-Danlos syndrome. So hyperextensibility of the skin and the joints are the hallmarks of Ehlers-Danlos syndrome. Next, Marfan syndrome. Abraham Lincoln perhaps had this disease. That so far I know, and it was reported. So there is a defective gene called fibrillin 1, and that is arachnodactyly. They are usually longer height. They are having a long height. Arachnodactyly means long digits. A normal hand, and Marfan syndrome, the fingers will be longer. Ectopia lentis means lens, the suspensory ligament of the eye, that lens will be weakened. So the lens will not be in proper place. There will be dislocation of the lens, hyperextension of the joints, and most importantly, aortic aneurysm at its origin. Can you imagine the fate of this child if he lives longer? At the origin of the aorta, there is aneurysm. If it ruptures, the case is gone. Next, Brenki's disease, typical kinky hair of the child, and this is a lysyl oxidase deficiency. Kinky hair, metaphyseal widening. The bones are not that wide in other individuals, but you see the metaphyseal widening, osteoporosis, and deposition of bone at the base of the skull. And this is nothing but the defect of lysyl oxidase. Next, ascorbic acid deficiency, vitamin C, SCARV, you have read it long back. So that causes gum bleeding, that causes fragility of the blood vessels, bleeding gums, subcutaneous hemorrhages, and there will be poor wound healing. That's why I told you during wound healing you do give vitamin C. The wound will heal much faster than otherwise. Next. So homocystinuria, accumulated homocysteine reacts with lysyl oxidase blocking cross-linking. So again you will see the skeletal deformity, vascular and ocular deformity. There is ectopia lentis. Next. Lathyrism. Many poor people take Khesari dal, and that contains beta amylopropyl nitrate. There is one variety called neurolathyrism which causes paralysis. There is another variety called osteolathyrism. You can see there is a damage to the bone, and this is due to cross-linking of tropocollagen and tropoelastin. Next. So what you have to remember about the diseases is this part, not that other part. This you must remember only, at least disease and the defects. Do not remember the features. You need not remember. You are not supposed to know at this stage, but you must be able to tell what I have written in basically highlighted in magenta color and the red color. Next. And these in a one tabular form I have written. Next. Next. So there are two other diseases called Allport syndrome and epidermis bullosa. These are not that important. Next. So to summarize, this is the last slide. This is a major component of BCM. You must remember structural protein, collagen, elastin, fibrin in one, specialized protein. Please move the this thing, video camera to the students' side. What they are doing. Let it be also recorded. So collagen is the most abundant protein. 28 types. I have told you specialized protein, fibronectin and laminin. I have told repeatedly. It's a triple helix structure, collagen, glycine, XY structure, complex biosynthesis involving hydroxylation of proline and lysine and extensive post-translational modification. Elastin, I told you in the difference that it lacks hydroxylysine, triple helical structure, sugars and glycine Y sequence, contain desmosine and isodesmoid complex not found in collagen. And it confers extensibility and elastic recoil